Good morning, students. Welcome back to online lectures. Now we are moving on to another topic, transcription. In the last class, we discussed about the features of RNA polymerase engine. Today, with the knowledge of RNA polymerase function and their structure, we have to understand how it involves in the transcription process. What is transcription? Synthesis of RNA using the DNA. That is, the information is transcribed into RNA form. So, again we will recall some of the points from the previous session. What are the functions of RNA polymerase? It is a single RNA polymerase performs multiple functions in transcription process. There are different points regarding to the functions of RNA polymerase. I am listed out. The first one, it sets and binds to promoter site, and it unbinds a short stretch of double helical DNA, and it selects correct ribonucleotides and catalyzes the formation of phosphodiester bond. Polymerization according to base pair rule. Here you can see the reaction. Ribonucleic acids in N number polymerizes with the nucleotide triphosphates, removing pyrophosphates, and the phosphate is added to the ribonucleic acid nucleotides. That is N plus 1 nucleotide. In this reaction, pyrophosphates are removed. The fourth one is detects termination signals, and it interacts with activators and repressor proteins that regulate the rate of transcription. Coming to what is promoter, to understand transcription, first we should know what is a promoter. A promoter is a region of DNA that initiates the transcription of a particular gene. Promoters are located near the transcription start sites of gene, on the same strand and upstream on the DNA. That is towards the 5' region of the sense strand. So promoters are located near the transcription start sites of gene on the same strand and upstream on the DNA. Upstream means the region left to the transcription start site. Promoters can be about 100 to 1000 base pairs long. Coming to RNA polymerase, as we discussed earlier, this polymerase involved in the synthesis of RNA in Escherichia coli. The polymerase have 5 subunits, 2 alpha subunits, beta and beta prime subunits, comprises a core enzyme. When this core enzyme associates with a specific factor known as sigma factor, together comprises holoenzyme. So the holoenzyme is required to recognize the promoter sequences on the DNA. Coming to transcription in prokaryotes, synthesis of mRNA occurs in 3 stages: initiation phase, elongation phase, and termination phase. Coming to initiation phase, RNA polymerase recognizes the promoter and starts the transcription. What is elongation phase? The RNA strand is continuously growing. What is termination phase? The RNA polymerase stops the synthesis and the nascent RNA is separated from the DNA chemical. So, coming to the prokaryotic transcription. Here we have to discuss some points. So, transcription is synthesis of the mRNA from the genetic material of the cell, that is DNA. Here the synthesis occurs in the cytoplasm. In the cytoplasm, RNA polymerase transcribes the genetic material and immediately, parallelly, transcription followed by synthesis of the protein known as translation. Whereas in eukaryotes, the synthesis of RNA occurs in membrane-bound nucleus, and now that RNAs are entered into cytoplasm and where translation process occurs. This does not see in prokaryotes. Why? Because there is no specific membrane-bound nucleus. The nucleoid is present, which is dispersed in the eukaryotic cell cytoplasm. So, transcription is known to be controlled by a variety of regulators in prokaryotes. Many of these transcription factors are homodietes containing helix-turn helix DNA binding motifs. So this gene expression, gene transcription is controlled by many regulatory proteins. They have the ability to bind to the DNA. See here the three stages are again initiation, elongation and termination. Here you can see the picture that is DNA occupied by RNA polymerase. This RNA polymerase occupies 60 base pairs length of the DNA molecule and it allows the transcription. Here we have to discuss many points during the initiation process. RNA polymerase binds to one of several specificity factors. One of that is sigma factor. In the last class we discussed sigma 70 and sigma 54. So they are known as specificity factors. So RNA polymerase can bind to the specificity factor sigma to form a coenzyme. In this form, it can recognize and bind to specific promoter regions in the DNA. What is the promoter region which initiates the transcription, which is found upstream from the transcription start site? The promoter regions contain some conserved sequences. Thereof, the sequence -35 region and the -10 region. Whereas -10 region is known as Rna box or Tata box. These regions comprises the core prokaryotic promoter. And here T stands for termination. Termination is nothing but this is the certain signals found distant from the transcription start site. When polymerase reaches that site, transcription ends. And the polymerase can recognize these sequences, especially sigma factor of the holoenzyme recognizes the sequences of -35 and -10 sequences found in the promoter regions. And binding of the enzyme to such sequence of the DNA forms closed complex structure. This is closed enzyme DNA structure is formed. Coming to what is about these sequences. What is Rna box? It contains 6 nucleotides. The sequence is TA, TA, AT. That means repeated sequence of TA, TA, AT. Located 8 to 10 nucleotides to the left of the transcriptional start site. So where they located? From the transcription start site to the left of that site, around 8 to 10 nucleotides are found. The initial basis of the mRNA, these are. And -35 region is second sequence nucleotide containing TT, GA, GA repeated sequence located 35 nucleotides to the left of transcriptional start site. To the left of transcriptional start site, that is -35 region. The DNA is unwound and becomes a single stranded. That is nothing but open complex is formed here in the vicinity of the initiation site, defined as +1. So whenever the complex is open and it starts to synthesize, that site is called transcription start site, and that initiated nucleotide is known as +1 site. The holoenzyme unbound DNA structure is called the open complex. Here you can see the picture of the strands, double stranded DNA, 2 helical strand, one polarity 5' to 3', another one is having 3' to 5'. They are complementary. Here the strand which is acting as template for the synthesis of mRNA is known as a template strand or non-coding strand. The above strand which is having polarity of 5' to 3' strand is known as coding strand. That is also known as sense strand. Whereas template strand also known as non-coding strand and anti-sense strand. Don't confuse with such words, na. You can remember template is nothing but which is involved in the synthesis of mRNA. The above complementary strand is known as coding strand. And see what is about upstream region and what is about downstream region. You can see the start point of the transcription region, where the transcription start site to the left portion is known as minus regions, -1. That is the first nucleotides left to the transcription site is known as -1 nucleotide. Next one is -2. Next one is -3. Next one is likewise. And the nucleotides right to the transcription start site are known as, are indicated with +1, +2, +3 in such a way. See here, come to the promoter region found in the upstream region. See the second figure here. The regions of -35 and -10. Here the -10 sequences are known as Tata box or Pigno box, found left to the transcription start site, and where we can see around 6 nucleotides TA, TA80 repeated sequence. And from these 10 nucleotides, 17 base pairs away from the Pigno box, another sequence that is -35, that indicates from the transcription start site the distance is -35 nucleotides. So thereby the sequence is called -35, that is TZ, TT, GA sequence. So these are the sequences recognized by sigma factor of the holo engine. Here this is the entire picture, you can understand easily. See the first one, double helical DNA, where the promoter region is found. So to which RNA polymerase binds and a closed complex formed. Later on, the RNA polymerase unwinds the double helical DNA at the promoter region and starts the initiation of the RNA synthesis. So after initiation, the sigma factor is no longer required to elongate the synthesis. Only the thing you should remember is for the initiation, for the recognition, only sigma factor is required. Thereafter that must be released, otherwise RNA unable to elongate. So thereby sigma factor detaches from the holo engine. Now the core engine beta subunit starts the polymerization and extends the RNA chain synthesis. The synthesis you remember always in 5' to 3' direction. In such a way elongation proceeds further. And finally, when the RNA reaches termination signals on the DNA, the RNA detaches from the DNA and it starts to translate into a protein. So here are the points you should remember. The RNA polymerases transcribes the DNA, especially transcription is achieved by beta subunits in the core engine, but they produce about 10 transcripts which are unable to leave the RNA polymerase because the exit channel is blocked by the sigma factor. So RNA polymerase initiates the synthesis, but the transcripts are abortive transcripts. Why? Because from the DNA, RNA is unable to displace because the exit channel is blocked by sigma factor. Thereby, the sigma factor must be released to release the transcript. Then only the elongation is proceeded. Coming to the elongation. Once the promoter region has been recognized by sigma factor of holo engine, the engine begins to synthesize RNA sequences. Sigma factor is released. This enzyme has no exonuclease activity. That means exactly it identifies the right nucleotide and incorporates it, but it does not have any proofreading activity. It is unable to move backward and check the nucleotide. So simply it starts to polymerize. So that's why error rate is more for the mRNA synthesis. Here you can see the picture how enzyme binds to the double helical DNA, to the promoter region of the DNA and starts the initiation, unwinding of the double helical DNA and synthesis of the RNA from 5' to 3' direction, initiation followed by removal of sigma factor followed by elongation of the RNA synthesis. Here the polymerization is nothing but addition of the nucleotides. Here the nucleotides are for the adenine uracil pairing, for the thymine adenine pairing, for the guanine cytosine, for the cytosine guanine. Always the chain RNA synthesis initiates with either ATP or the GTP. Whereas for the uridylated triphosphates, UTPs and GTPs are not favorable for the initiation of the synthesis. Coming to the termination. So in such a way the process elongates until the termination signals are present on the DNA. The transcribed region of the DNA template contains stop signals. Prokaryotes have two classes of termination signals. One that relies on a protein factor called Rho factor, that is known as Rho dependent termination. The other one is Rho independent termination. Coming to Rho dependent termination. It is signaled by a sequence in the template. That means a special termination signal is found on the DNA, which is about 40 base pairs in length and are inverted repeats. There are inverted repeats. These signals are recognized by a termination protein that is called Rho factor. So this 40 base pair length of inverted signals recognized by Rho factor. What is the Rho factor? Rho protein is a ATP dependent RNA stimulated helicase. Nothing but this is RNA directed helicase. It uses energy. So which binds to the signals. Thus RNA polymerase cannot move further. So it dissociates from DNA. that disrupts the nascent RNA-DNA complex releases nascent RNA. So this is the picture showing the termination. Rho protein binds to the signals and the binding of the Rho protein does not allow the movement of the RNA polymerase and it breaks the bonds between the RNA-DNA hybrid and allow the release of the nascent RNA. Coming to Rho independent mechanism. This is most independent terminators have two distinguished features. This is of again two types. One is palindromic GC rich region which is followed by an anti-rich region. What is palindrome? Palindrome is nothing but it is a sequence which can read same backwards as forwards. This RNA transcript of this palindrome is self-complementary sequences, permitting the formation of hairpin structure. So this complementarity of the RNA allows RNA polymerase do not move further. Second one is the highly conserved string of A residue, that means repeatedly A series on the DNA, that is transcribed as uridine. So the bonds between A and U are very unstable. So thereby the nascent RNA easily displaced from the RNA-DNA hybrid. And you can see the picture here. The first feature is the consisting of palindromic sequence. You can see the sequence, two sequences representing. One sequence is nothing but the mirror image of the opposite sequence. When during the transcription, the palindrome itself has transcribed. So the transcribed sequences undergoes the self-complementation of the RNA. That leads to formation of hairpin that do not allow the movement of the RNA polymerase and thereby displaces the nascent RNA. And next feature is repeated A series of the sequences. A is transcribed into U. So the repeated A-U binding is more unstable. So easily the nascent RNA can be displaced from the double-stranded DNA. So that's all about the transcription. Thank you.